Hello, my name is Hannah Mirshahi, and I studied pathogenic variants in the TGFβ signaling pathway that increase the risk of thoracic aortic aneurysms. Thoracic aortic aneurysms, or TAAs, occur when the enlargement of the aorta causes a tear in the aortic wall, which can progress into the lethal rupture of the aorta. Risk factors for TAAs are age, being male, and smoking, but there are also genetic risk factors involved. The TGFβ signaling pathway is essential for maintaining the integrity of blood vessels like the aorta. Therefore, one can speculate that defects in this pathway may put individuals at risk of TAAs. We hypothesize that genetic factors that may lead to loss of function in 4 proteins in this pathway may contribute to TAA. These genes are SMAD3, TGFβ2, TGFBR1, and TGFBR2. To test our hypothesis, we used data from the MyCode cohort, where genetic data for 175,000 participants is linked to their electronic health records. Our approach was to identify variants in these 4 genes and determine if they are linked to increased risk of TAA. Here's a table summarizing the variants we found in the 4 genes. We found some protein truncating variants, which alter the structure of proteins, as well as amino acid changing variants, and some of which were novel. Several of these variants were also listed as pathogenic in ClinVar, which is a website where researchers link variants to clinical phenotypes, and we found a high prevalence of TAA amongst pathogenic variant carriers in the cohort. We also determined the odds of disease for each type of variant per gene, modeled here in these forest plots. We found that the risk for TAA increased greatly amongst pathogenic variant carriers in all 4 genes, and amongst protein truncating variant carriers in the SMAD3 gene. Using relatedness data, we also constructed family pedigrees, finding variants of unknown significance that co-segregate with the disease. This finding suggests that we may be able to classify these variants as pathogenic in the future, and we can use knowledge of carriers and case history in the family to take preventative measures for carriers who have not yet had an aneurysm. These findings can be used to enhance the prevention of aortic rupture through identifying at-risk individuals. And on the larger scale, our data can also provide some new insight into the progression of aortic ruptures and how to prevent them. Thank you.